briefly, let's look at enzyme assay. Whenever you hear enzyme assay, we are talking about the laboratory techniques or laboratory procedures that is used to determine the activity of an enzyme. So whatever method you decide to apply in the lab, that will enable you determine the activity of an enzyme, that is how fast or how slow an enzyme is working. That method is described, or the process is described as an enzyme assay. They are very important in determining the kinetics of an enzyme. They are very important in inhibition studies. I hope we know that most drugs that we take today are enzyme inhibitors. So the study of this assay of enzyme also helps in drug delivery and discovery. So they act as a way of measuring how fast an unknown enzyme can convert substrates to protein. That is a way of measuring their velocity. Okay. Now, in enzyme assay, it measures one of these two things. Either it is measuring the way the product is appearing, or it is measuring the way substrates are disappearing, because substrates must be converted to products. So you can use both ways to measure the activities of an enzyme. In other words, if you're making a plot of your product formation per time, you're going to get a positive graph. If you're measuring the disappearance of substrate per time, you're going to get a negative graph. Okay, now that is when you're plotting the graphs. But the summary is that you can use the way the substrate is disappearing to determine the activity of the enzyme, or you can use the way products are appearing to determine the activity of the enzyme. Okay, now enzymes are usually present in very small quantity in biological fluid, therefore they are not directly measured. Okay, so because of the quantity, the concentration of enzymes, it's difficult to measure the quantity of enzyme. Okay, so instead what you do, you measure how it's working. Instead of measuring the amount, you're measuring how it is working. Now, the most common, or they are commonly measured in terms of their catalytic activity. Okay, so and the unit of enzyme activity, you measure it in kcal, but kcal is a very big quantity. Okay, so later brought down to micromole per minute, even though you measure the kcal is 1 mole per second, but the more practical unit could be enzyme units, that is U, which is equal to 1 micromole per minute, not seconds. And this 1 micromole per minute is corresponding to 16.67 nanocatals. Okay, so that is why, because catal, the amount is, the quantity is always very high, so they decided to make use of micromole per minute, or so on and so forth. So you can see that you're looking at quantity per minute, which means you're studying velocity. Now, specific activity of an enzyme is another common unit that you use. Okay, that is the activity of the enzyme per milligram of the total protein. We mentioned that in the other lecture. Now, types of assay. There are two groups of assay, or two types of assay. We have the continuous and the discontinuous. Okay, and each of the continuous or discontinuous, you can have the direct or the indirect stroke coupled in both of them. So in a very nutshell, somebody can simply say there are four groups or four types. That we have direct assay, we have indirect assay, we have continuous, we have discontinuous. But in a more clearer view, you should see it from the point that we have continuous, we have discontinuous, okay, and each of them, they say direct continuous and indirect continuous. Then there is a direct discontinuous and there is an indirect discontinuous. So these are the types of enzyme assay. Now let's look at continuous assay. We say continuous where the assay gives a continuous reading of activity, multiple measurements, usually of absorbance changes are made during the reaction, either at specific time interval, maybe 30 seconds, 60 seconds, 1 minute, 2 minutes, and so on, or continuously by a continuous recording in the spectrophotometer. These assay are advantage over fixed time method because the linearity of the reaction may be more adequately verified. Okay, now in this continuous method, we just say you take the reading continuously. Okay, you don't need to stop the reaction. You allow the reaction go, but you take your reading with time. So that's why it's called continuous. And this might have given you an idea of discontinuous. You have to stop the reaction, take your reading. But that is not in continuous. You continue the experiment and take your reading with time. So the methods that you use in continuous varies. And this method, you may use spectrophotometric method, the fluorescent method, the polarimetric method, sampling method, manometric method, electrode method. Now, a good enzyme assay must have the following. It must be simple and specific. It must be rapid. It's something that you can do quickly. It must be sensitive, which means you have to use very small samples, and it has to be easy to use and also economical. Okay, well, you can actually see the use of spectrophotometer. I'm not going to teach you that, but you have to look at the advantages. And the idea in using spectrophotometer, for instance, in this equation, fumarate is being catalyzed by fumarate hydrates to malate. And we are saying that fumarate absorbs at 300 nanometer, but malate does not absorb. So in this case, what you do is that you check to determine the activity of this enzyme. You check the disappearance of fumarate. Okay, you measure the disappearance of fumarate as time goes on. You allow the experiment to continue, and you continue to measure. Now, also, you have hypoxanthine when it is being acted upon by xanthine oxidase to form urate. You see urate absorb, but hypoxanthine does not absorb. Urate absorbs at 290. So you begin to measure the increase in absorbance of urate by at 290. Okay, so this will help in this. That's how it's being determined now. But that is for um using the product of the reaction. Now, sometimes both the substrate and the product do not absorb, but some of this reaction they might be making use of coenzymes and cofactors. Okay, now the coenzyme might be absorbing, but the substrate and product does not absorb. So in that case, you can measure the changes in absorbance of the cofactor. Okay, for instance, or the coenzyme rather, before I say cofactor. For instance, the NADH absorbs absorbs light at 340 nanometer, but NAD does not absorb light. So in the reaction which involves lactate dehydrogenase to convert pyruvate to lactate, you can measure the rate at which NADH is decreasing. Okay, because without NADH that reaction will not go, and without pyruvate lactate will not be formed. So when you have your pyruvate, your enzyme, and your NADH, you begin to measure at 340 nanometer. What you're measuring is your NADH, which is now being depleted to form NAD. So it is another form of continuous assay, and it's quite direct because you're acting directly on the enzyme. Okay, now we also have coupled reaction. In coupled reaction, let me just go straight to this. In coupled reaction, it means you're coupling that reaction to the one that maybe the product, the enzyme and the product, cannot do not produce light or does not absorb light. In a case of citrate to isocitrate. Okay. Now, if you're interested in in determining the activity of, say, aconitase, okay, our citrate does not absorb, our isocitrate does not absorb. So what we do is that we couple this reaction to the conversion of isocitrate to alpha ketoglutarate, in which the enzyme that actually does the job of this conversion is um, is isocitrate dehydrogenase. Now, what we now do in this case is that you're going to put your citrate, you're going to put your aconitase. Okay, then when you put it, you're going to add your isocitrate dehydrogenase, then you're going to add your NAD, and you begin to monitor the production of NADH. Okay, now the sense there is that if aconitase is not working at a fast rate, it's not going to produce isocitrate at a fast rate. Okay, and if isocitrate is not produced, then isocitrate dehydrogenase will not perform. Therefore, NAD will not be converted to NADH. So you see, in this method, these reactions are coupled to each other. So you are measuring NADH indirectly, okay, that's why it's an indirect method, as a function of the aconitase. Okay, so we see so many coupled reaction. Take note of them. ATP does not absorb, but NAD does. So you use, if you want to actually check the activities of pyruvate kinase, you actually begin to couple that reaction to lactate dehydrogenase. Okay, so that you measure your NAD, which can absorb in all the components of this reaction. So that's, we have different methods. Efflorescence, I want you to just read these ones up: calorimetric method, chemiluminescence method, manometric method, and so on, electrode method, polarimetric method, and so on and so forth. So in discontinuous assay, like I mentioned earlier, is an assay that you have to stop the reaction before you take your reading. That's just a difference. Radiometric method, you use it to monitor reactions that have to do with radioactive isotopes. Okay, so and so on and so forth. There is no much thing to do in enzyme assay, but it is something that you just have to study in case. Anyway, thank you very much. The question is still through our channel, and don't forget, it's very important you go through all of them.